Well, good afternoon, everyone. My name's Carl Larson from RMCG. Today, our webinar is focusing on soil-borne diseases and more specifically managing the risk of those. We're joined by a couple of guest speakers, Michael Rettke from the South Australian Research and Development Institute over in Adelaide, and my colleague Doris Blaesing from RMCG down in Tasmania. So we've got the tri-state approach, Victoria, South Australia and Tassie all in one, which is the beauty of these types of sessions. Now today we're going to hear from Doris first, just to really set the scene. What are some of the key issues and principles in managing soil-borne diseases and some commonalities across crop type before we get into, I guess, the main part of the presentation with Michael Rettke around the role of soil DNA testing in actually managing risk pre and post plant, which he'll go into in a bit more detail, and then plenty of time for questions throughout, and also at the end we've got a designated period of 15 to 20 minutes for Q&A too. Now, if you haven't joined us on one of these webinars before, just a quick overview of what you'll be seeing on your screen. Obviously our PowerPoint and webcams are shared so you can see what we're looking at and presenting on, but you'll see that there's a control panel on the side there that enables you to see who else is on the line in the attendees panel, and also a section there just to type in some questions which will come directly through to me. Feel free to type those in at any time, and also at the end where you've had that light bulb moment or want to seek some clarification on something, they can be put in at any time. There's also a section there on the pane called handouts. Now there's 5 PDFs there on relevant content to today's webinar that we've put together as a bit of an education package, both on managing soil-borne diseases more broadly, but some specific issues in there like damping off in spinach and pythium in carrots. Also some broader guidance around biofumigation, which Doris will touch on shortly. Now, today's session is being run by the Soil Wealth and Integrated Crop Protection projects, which is really focusing on getting the R&D that's done through the levy and co-funded by Hort Innovation and the Australian Government out onto farms around the country. That's been running in the first phase since 2014 for 3 years, and we've just recently ticked over into phase 2. Many of you might be familiar with some of the demonstration sites around the country. We've now got 9 new sites operating in the key growing regions, which are complemented by a number of different events like today's webinars, but also face-to-face farm walks and masterclasses, as well as some resources which are available in your sidebar that I mentioned before. Before we do get started, it's always helpful for us to know who's on the line. And I'd like to just launch a quick poll now, which you'll see come up on your screen, which is just asking a very quick question. How much experience do you have in actually managing soil-borne disease in vegetable crops? We'd like you just to quickly now and anonymously just select one of those 4 options, either lots through to none. That just gives us a bit of an idea as to where people are up to and how quickly to move through some of that content. So I can see now that most people have voted. So I'll close that off and share the results back with everyone. So we've got the predominant cohort there with a bit, 65%, and about a quarter of us with not much. So look, the aim of these lunchtime sessions is to really make sure we bring people up through that rank. If you're sitting at not much, hopefully you know a bit or lots by the end of it, and that just helps Mike and Doris pitch some of their messages. Okay, so without further ado, I'll hand over to Doris. Doris, I'll just share your screen. That should be coming up with you now. And over to you.
Okay. Good afternoon all. I would just really, as an introduction, show some of the results we got looking at soil-borne diseases in vegetable crops, just assessing, what are the big, not the big 5, but certainly a few big ones, and what are the commonalities between crops So I don't want to read the slides to you, but just keep an eye out for the same diseases appearing over and over again. But certainly in brassica crops, the big one is clubroot, and quite a lot of research has been done on how to manage clubroot. If you need more information, you should let us know, and we can make sure that is on our Soilborne's website. And also, Michael will talk about clubroot in his presentation. The major soilborne disease in carrots, as you can see in the images, is cavity spot. It's still a hot favorite, and also nematodes. The nematodes will and Pythium can cause forking. The main Pythium we have in Australia is Pythium sulcatum. It does like it hot. I will, in the second part, talk a little bit about management and managing risks. So we just here have a bit of a rundown of what's actually out there. A rather new crop is, the baby leaf spinach, baby leaf lettuces, and the big issue there are damping off diseases. And they're not only one, there's a whole disease complex. And from that point of view, difficult to control with chemicals. You could try to control Pythium and get more problems with the others. So there's still a bit to learn, and our soilborne disease project, which have been running parallel to soil wealth, has looked at specific management for spinach diseases, and I think we've got some fact sheets on that as well on our website. You can see lettuce suffers from the same problem, but also one of the rather big ones is the big vein disease, which is associated with a fungus and a virus. Chilies, we've got a big problem there with, again, nematodes, and you can see the difference between the lesion nematodes and the Rootknot nematodes, which also affect a lot of other crops. And again, our damping off turns up again here as well. Beans, widely grown in Queensland, in Tasmania, again, Sclerotinia, again, damping off, but we also have southern blight, which is the same pathogen as we've saw previously in capsicums, and then the beans have their own charcoal rot as an added problem. A lot of issues in alliums, so leeks as an example is pink rot, and basal plate rot, again a fusarium that we've seen as a damping off disease previously. So this is just a really quick overview. We've done quite a bit of work to get to this reasonably simple list of the major diseases in our major crops. And prevention is really, there is a lot we can do apart from trying to use chemicals, which often are not as successful as one wishes, given we're dealing with soilborne diseases, and the soil is a really difficult medium to control diseases. So the first is site selection, and Michael's presentation will talk about that a bit more, how we can do that smartly. An important one is to actually have tolerant and resistant varieties. We often forget how important that is. Rotation is important, and really our issues are now that because of economic pressures to have to grow the same crop over and over again, so the other option, if you have a lack of rotation, to really look into soil health management, organic matter, preserving organic matter, and with that biological diversity, making sure there is no compaction, making sure there is good drainage, and as an option of missing rotation, try to grow diverse cover crops and biofumigation crops. Again, we've got quite a bit of information on our website on biofumigation crops and examples from our trial site. So please have a look. And if you have questions, we are there to talk to. We're not just some faces on a webinar. One really important issue for dealing with, especially damping off, is fast crop establishment. So if crops are planted under challenging conditions, you will have more issues with soilborne diseases. Soilborne diseases all like high soil moisture. So by actually having good drainage, it's already a winner. And any soil that is compacted through to tillage and management will not drain well, and you will get problems. There's also some weird and wonderful products that might help in fast crop establishment. We can't really go into detail, but again, if there's interest, we will provide more information on our website. on, and we're still working on that, on products that might help biological activity in crop establishment. Another really important part is having clean seed. It is worthwhile getting seed tested for pathogens because a lot of pathogens can sit in and on seed. Also, it's worthwhile if you get transplants to actually visit your nursery and make sure they have the right procedures, and you get plants that are just ready, they're not pot bound, and they're not stressed already when they come to your farm. Nutrition is a really important one, and excess nitrogen doesn't really only mean excess nitrogen as such, but unbalanced use of nitrogen without having the required calcium and trace elements and potassium available. So again, we've got quite a lot of information on soil testing and plant testing and managing nutrition, and we're actually running a masterclass on that topic in October in Brisbane. So watch that space and sign in for that. Now stress is as bad as for plants as it is for us. So any heat, cold, or any other stresses will make it more prone to soilborne diseases. There is a little bit of microclimate manipulation properly possible, especially if you use plant spacing or canopy type. In beans, the canopies that seem to get floppy are certainly more prone to sclerotinia. Hygiene, an important one we tend to forget, but also important for biosecurity. Water testing might be a good idea if there's quite a few diseases being transferred by contaminated water. Pesticide programs, that's often the way we try to control diseases. If you do not do other risk management, it's probably going to be a hard one to control diseases, soilborne diseases with pesticides. Fumigation is still used, and sometimes people don't get away without using it. However, it should be a last resort, and we really should try to avoid fumigation if possible. Now we're coming a bit closer to Mike's presentation. Make sure you understand what the risk is. House crops in the area-wide management might be quite important. So it's not only what you do on your farm, but what your neighbors do is important. Weather monitoring and forecasting, we're getting better at that, and there's a lot of systems that help us with forecasting. They're mainly still for leaf diseases, but there will be work on soilborne diseases as well. And really, the last thing and the important thing that Mike will now get into is the pre-plant soil tests and seed tests we cover, not here, but the pre-plant soil test. So I hand over to Mike.
Right, thanks for that, Doris. Mike, I'll just transfer over to your screen. And we'll bring up your presentation. And just a reminder too, you can type those questions in to the control panel to come through at any stage. I've just got one there that I might hold over until the Q&A session and use that segue that Doris has given us to move on to Mike's session. So Mike, over to you, thanks.
Okay, we're all right to go.
Yeah.
Good afternoon, everyone. Thank you for joining us this afternoon. I'm going to take you through, I guess, some of the testing that we have, but also the broader role of what soil DNA and DNA testing in general can do, both in terms of some diagnostics, but what mostly the technology has been developed for is assessing the risk of soilborne diseases. Now, with soilborne diseases, Doris went through quite a list of them. There's some that cover a number of crops, and there's some that are specific to certain crops. But you really need to know which ones are most important in your situation, your cropping system, and your environment. And it's really difficult to assess that just by looking across your paddock. Now, what we've really developed is another way, a way of assessing that, which I will go into. Now, I will cover some of the other aspects, as I said, of what you can use pathogen DNA testing for outside of soils as well. Now, all of these things, soil DNA testing or DNA testing is just part of a great big picture. And you may notice down the bottom, I've got Barbara Hall's group. which is actually horticultural pathology or traditional pathology, and we've also got Mason Carney in that group. So it's very important that you support all of these findings and technologies with actual traditional diagnostics. But then we've got Alan McKay's group, who is the head of our molecular diagnostics unit. So I think we should probably start by, before we launch into talking about risk assessment or how we may do certain things, I think we need to sort of set the scene for people of what actually is pathogen DNA testing. What are we actually doing? Like, it's a black box technology, some people might say, but what we're doing, or the way that we've developed the technology, is we're measuring the total level of the DNA in whatever sample that we're looking at. Now, it's going to pick up everything. It's going to pick up on that slide there. You've got some J2 Meloidigan phalanx, you've got eggs, you've got spores, you've got long-term resting spores, but you'll also have hyphal growth. Everything that's in that sample, we are basically measuring what the total level of that, and that is all contributing, well, not all, but most of it will be contributing to your inoculum level and potentially your disease risk. Now, the second thing of the technology, the particular technology that we're using, is that it is designed for a specific pathogen. So we are looking at usually to a species level. So if we test for Rootknot nematodes, we'll be testing for a particular Rootknot nematode, say Meloidigan phalanx. So we go down to that level. Now, that gives you very detailed information on what the breakdown of a particular, say, nematodes are, which is more difficult to do by traditional methods, but it also means that we only pick up things that we have specific tests for. So that's something you have to bear in mind when you're using this technology. Now, what can we apply it to? I guess we specialize in doing soil samples, so we can do soil samples of up to 500 grams per time. So that's a sample bag that you'll see, right? So we can quantify the specific pathogens in that soil sample. But we can apply this technology to plant tissue. So there on the slide, you've got some pictures of some knobs and bits and peels of carrots. So we can actually quantify what the pathogen levels for the tests that we've got in that sample, right? And we can do that probably up to dry weight, 50, 70 grams, right? So you can get a pretty good sample across a paddock to actually look at that, or you may just do an individual symptom. So you can actually just test the individual symptom, right? Which we do slightly differently. Now, on the last one, you can apply this technology to most substrates. So you can do, and what I've got there is some brassica seedlings that have come out of trays. So we can actually combine them together up to nearly 500 grams and actually run to see if there is any pathogens in that tray, right? So that's something we can do. It's a potting mix. We actually have to do it quite differently, but that can be done, and we do that for research purposes, because you really need to be taking account of all potential disease sources or sources of inoculum, right? But I'm mostly going to concentrate on the soil side of things with pre-plant risk assessment. Now, as I said, our tests are really designed for specific pathogens, right? So I'm not going to go into all of the detail of all the species because that'll take some time, and I've only put up the list of numbers of tests for those that are relevant to vegetable crops on this slide. We have some 90-odd tests that we can access and use, and if you're interested in that, you can go to our website as listed there, or just type in molecular diagnostics or predictor. Will generally get you to that website quite quickly. So, and there you can access a full list of the tests and the panels that we can run. But specifically to vegetable crops, I'll go through, say, the nematodes. We have 5 different tests for Rootknot nematodes, which gives us very good coverage of Rootknot nematodes in Australia across pretty much all areas, all crops, right? So we've got very good coverage for that. For root lesion nematodes, we have 5 different species tests relevant to horticulture. We do have a couple of broadacre ones as well. In the southern cropping regions, we have pretty good coverage. As you move further north, there are a couple of species that we don't currently have tests for that may be important for certain crops. We also have a stem nematode test and a clover cyst nematode test as well, which can be applicable to certain crops. Now on the bottom, which has been used a bit in broadacre, but not so much in horticultural situations, although we are starting to investigate it, is we have a series of 15 what we call free living nematode tests. So you're basically looking at your beneficial side of things to give you the balance. Now in broadacre cropping systems, it can give you a reasonable indication of the status of that cropping system. We're not quite at that stage in horticulture, but we've certainly been, in our recent work in brassicas and carrots, we've been running those tests to see how useful that may be and to see whether it gives an indication, I guess, as an overall soil health indicator. If I move on to the fungi, the main areas that we have is Rhizoctonia, so Rhizoctonia solani, which is one of the major Rhizoctonias across a range of crops. So we have 6 different AG groups for that, and it's important that you go down to the AG group level because some crops are only affected by certain AGs. So if you're going to assess risk, you actually have to be operating at that level. For instance, for brassicas, it might be AG 2.1, AG 4, right? Whereas onions, it would be AG 8, right? So we have those. So we have reasonable coverage of those for most of the crops. There are a few exceptions, and lettuce would be one of those, which would be an exception. Now, the Pythium tests, we've actually designed 2 new tests, which I'll talk about a bit later, which are the ones Doris mentioned earlier for cavity spots, so Pythium sulcatum and Pythium violae. But we also have 2 other what we call clade tests, which cover a whole clade of different species. Now, those tests are not probably as useful because it depends on, you don't actually know what the species mix is there, right? But they can give an indication and they can be useful coming out of crops to see how much Pythium is in that crop. There are 9 other tests, which I won't go into detail today, but we're happy to talk about those always for other crops. But they're things like Macrophyllina, Verticillium, Aphanomyces, also the pathogens that cause blackleg. So, we have got a reasonable range. We have also got, recently we've got some sclerotinia tests, which are both broad and specific. So we've got 2 tests there. So we don't have a lot of experience with them yet in vegetables, but they will get used in the near future, particularly for things like, lettuce, brassicas. So in the bacteria, we don't have a lot of coverage in bacteria. It's not one of our key areas. We do have a Streptomyces test, which is relevant to some vegetable crops, more relevant in potatoes, but is relevant to some. And I think Doris mentioned Streptomyces earlier, or scab symptoms. And obviously the other one, which we've only developed in the last year or so, is the pathogen that causes clubroot, so Plasmodiphora brassicae. So, and I will speak a bit more detail about that. That and carrots are the main ones I'll talk about today. So what would we apply? Like, where are we going to actually use this? What are we going to do with it? You can think about 2 things, and I guess the first one would be investigative. You've got a carrot paddock there, you've got those symptoms. What are they? You can use standard diagnostics to do that, and you would do that. You dig them up, they've actually got quite deep lesions on those carrots. As it turns out, they're Rhizoctonia solani AG 2.2, right? So a DNA test can quantify that and tell you just how much of that is in there, and you can look wider and do a post-mortem. It's only part of the diagnostic picture, not the whole diagnostic picture, but it's certainly useful to have those specific tests at your disposal. Now, that's great, doing post-mortems. You can do a post-mortem with a rotary hoe, unfortunately. It's, that's not the place you want to be. I guess what we're looking at is how do you not have the post-mortem, right? We're looking at technologies that can help you to assess the risk up front so that you can make decisions that will potentially reduce that risk. You can't reduce the risk completely, but the more information you've got about what you're dealing with and what risks are, the better you can make your decisions. Now, I'm going to actually use, because I'm actually going to use some examples of the services that we've already got before I actually go on to talk about carrots and brassicas. So SARDI already provides pre-plant risk assessment services in potatoes, in cereal crops. In cereal crops for a long time, potatoes since 2013. We also, on request, we will do pre-plant risk assessment testing for onions. Now, this is where we're wanting to get to with the vegetable crops, which is what I'm going to focus on today. But just so that you know how the technology works, it's one thing to develop a test for a pathogen and then to make sure that it's worked, make sure it's specific, make sure it's quantifying, all those sorts of things. But you actually have to take it out in the field as well to actually determine, is it providing useful information? Not all pathogen tests are equal in this regard. That depends a lot on how low the thresholds are, whether we can detect down to those thresholds. Also, how much the environment plays a role, particularly for low threshold pathogens. Phytophthora is an example of that. So you have to go through a process of working out how useful they are. They're always useful for diagnostics and other things and assessing trials, but as a pre-plant risk assessment tool, you go through a process of looking at a lot of sites across a lot of environments and a lot of conditions with a lot of inoculum levels. And that's what the graph you're looking at there for powdery scab of potatoes is. So just to explain it, if you can see where I'm pointing the mouse down the bottom there, that's basically what we're measuring. So we're measuring the DNA of the pathogen, which is Spongospora subterranea in this case. So we're measuring the total amount of DNA in the soil prior to planting, and we're getting a level across here. Now, to actually validate these tests, what we've done is we've gone out and we've assessed what the incidence of powdery scab is on all of those paddocks at the end, or sites at the end of the day. Now, what you'll get, it's not a predictive test, okay? It's all about probability, because if you have a high inoculum level in a paddock, which is the case out here in this red zone, it doesn't mean you're going to get the disease, because that depends on the variety that you're growing. It depends on the conditions, when it was planted, a whole series of things. So you'll get a mix from high incidence down to no incidence, right? What you don't want in a good test, which we regard this as a good test, is you don't want high incidences where you're not detecting it or detecting it at low levels. So that's basically what defines a useful test for pre-plant testing. So now the second thing I'll add here is part of that process, a lot of effort goes into how you sample, and that's a very important part of getting good results with this type of testing. You have to sample using the protocols that we've established over time to give a good risk assessment. If you don't sample properly, you can't expect, because ultimately it's what's in this bag that determines what result you get. So you've got to get a good representative sample into there, and we've spent a lot of time working that out. So if I move on to where we're at with vegetables, because we've only really been in this a couple of years, moving into this space, whilst we have done testing in vegetable crops before that, but we've made a concerted effort in the current project over the last couple of years. Now, one of our key targets has been clubroot of brassicas. Now, as you can see by the shot there, clubroot of brassicas can be quite a devastating disease and can result in complete crop failure when inoculum levels are high and conditions are favourable. Now, so what we're doing is we're trying to use our pre-existing knowledge of how things work to get decent amounts of graphs and data that actually gives us confidence in a test. Now, you would expect this test, because this pathogen's actually a very similar pathogen to the powdery scab of potatoes, for which I showed the graph before. Now, we have data, and we collect data from both little trials where we have plots within the same field, and we look at different levels of inoculum within that field, and then we go back and we look at incidence, we go back and look at severity. So this is one of the graphs out of that. This is a graph where we're actually looking at the lower end of the inoculum scale. So we're not dealing with huge amounts of inoculum of this particular pathogen. And you can see in general, as you move up, the incidence gets higher. And there's bar 1 point, it's pretty good at the low end. Now, we also have a lot of trials out of commercial paddocks, right? And this is done within South Australia, it's done within Tasmania. We also have sites in Victoria, right? So we're compiling all that data together. But if we looked at our general South Australian data, we've actually got starting levels that are 10 to 100 times higher than anything that's in this trial. And they're going to put a lot of points, and they are putting a lot of points right at the top of this graph, right? So when we look at it broader. So in general, this test looks pretty promising, right? So, and we're looking to progress it. On the top, you can see this is what we'd call a lower incidence. You've got some major clubs in there, but you don't have completely clubbed roots like you can when you're looking at a situation like this, which had much higher inoculum levels than anything on that graph. Right. Now, at the same time as looking at these, we're also using all of our other pathogen tests. So, for instance, for brassicas, we're also simultaneously looking at what our Rhizoctonia levels are doing for the ones of interest. We're looking at nematode levels, that we have at our disposal. So we're running all of those tests at the same time. So we may get data on stem canker or wire stem or just general yield reduction from nematodes. So we collect that data at the same time. Now I'll move on to the other crop that we're focusing on, which is carrots, which is the crop that we've developed 2 new tests for, so Pythium sulcatum and Pythium violae, targeting cavity spot, and I'll actually show some symptoms of that later and discuss some of the diagnostic type use of this technology. Now, the violae test it's on the other end of the scale to the previous one. It's a challenging test because it has the capacity to go from fairly low levels to high levels, right? So you're going to be looking at relatively low initial thresholds potentially for that. Now, for Pythium violae, we don't have a lot of detections, and as Doris said, it's the lesser common of the 2 in Australia. But we have now, I guess, identified some sites where it is, and we need to follow up with those to generate more data. Now, we have found it in other places, but we've not really found it at levels that would be commercially worrying. Whilst it's there in the final crop, it's not really been at a level of concern. Pythium sulcatum is far more common in the areas that we've been. And I guess what we're finding is it is very environmentally driven, which is what you would expect. And it's also by the variety susceptibility as well. But it does appear to be giving a good indication of when you detect it, that it's there and has potential. So we need to further investigate that with some more sites, but we are getting there with that. But whilst we're doing similar process, we're doing commercial fields where we're looking at them on 1 hectare scales and in paddocks bigger than that, right up to considerable size pivots, right? But we're also doing plot trials, which is what the 2 photos are on this slide as well, where we're looking at more specific situations where in this case there's only really 2 pathogens operating. So there's Pythium sulcatum and the other one is root lesion nematode, Paralingus thorni. So if we look at that, you can see that sometimes, whilst this is known as a carrot pathogen, hence why we're testing for it, it's not something that you're going to visually see a lot of symptoms of in the crop, and it can be just taking yield away. So I guess what we've seen in this particular trial site is an indication that this root lesion nematode is potentially reducing yield. Now, we have actually got a lot of data out of this site, so we've got soil nutrition data, we've got some monitoring of temperature levels, we've got monitoring of soil moisture levels, so we've actually got quite a lot of data to put together for this. But eventually we'll put all that together, and I guess we're going to investigate how much that root lesion nematode and how much some of the other soil nutrition factors are playing on this situation, right? Because nothing really ever works in isolation. You have to treat these things as an overall system. And you can clearly see in this trial that some soil chemical properties are more conducive to one of these pathogens, whilst other properties are conducive to the other. So you get a lot of information out of doing this type of work when you've got both nutrition data and soil pathogen data. Now, I'm going to move on to staying on the nematode theme, right? We're not going to try and reinvent the wheel here, right? of all the things that you can look at, because if you're looking across that paddock we showed earlier, it's very hard to know what pathogens are in there. You might go out there and look for weeds or something like that that may carry the pathogen, or you'll have past history and all those sorts of things that you can base into your decision. But nematodes is actually an area where pre-plant risk assessment using traditional nematode counting models is well established. Okay, so there's a long history of that. There's a lot of knowledge about nematodes. There's a pretty reasonable knowledge about thresholds for certain nematodes. And what we're really trying to do is A, calibrate our tests against that, but also B, because we're very species specific in our testing, to refine that knowledge a little bit and make better use of our capacity to easily identify the species mix for root lesion and root knot nematodes and the impacts of that. Now, as I said, it is quite established. Like, for instance, if we look at protected cropping, which is the graph there, we've just got a range of levels. So, you know, you've got a range of levels within that system. If you plant cucumbers into that system at some of the higher levels in there, then you could well lose that crop, right? That's because high pre-plant levels, young seedlings, very susceptible, right? As you get towards the end of the crop, they will often build high levels, but the plant can tolerate a lot more by then, particularly things like capsicums. But it's because it's a well-established knowledge in this area. As I said, what we're really trying to do is build on and make it better with DNA technology, right? So as another means of doing it, and the beauty of it is you can also do pathogen tests at the same time. So on the same soil sample, you're getting all of the tests done in one sample. The question is, okay, I know something about what my levels are, right? I might also have ground truth that against what I know of past history and anything else I've seen in the paddock. What am I going to do with this? Well, that really is dependent on the crop that you're growing and the options that you've got available for that and the major pathogens which are going to cause disease that you're dealing with. For some crops and pathogens, there aren't a lot of options. One thing that is generally true for these things is you need to know up front what they are, because you really need to be starting to manage these situations well before you plant, right? So you do need to have some sort of a risk assessment, whether it's using the sort of technology we're talking about today or whether it's just past history, etc., which Doris talked about earlier. Now, what might you do? So you get a high level for root knot nematodes. I think we talked earlier before that, well, that can result in basically a crop failure, right, for some crops. So, I mean, in a worst case scenario, you get high results. Unless you do something about them, you don't plant in that situation until you bring that to a situation where it can be managed. In other words, it's no point planting it into an unmanageable situation, right? But a more common use is, say, for the clubroot test. We move down to the next one. We're scheduling, right? You be planting your brassicas every week of the year, right? So you know that clubroots are the risks of clubroot are going to be much higher in the summer, hotter months. So if you know which regions of your property have got high levels of clubroot, now you're going to know some of that from past history, but you also can get some surprises. So if you've done some testing to do that, you're going to make decisions about what regions of your property and how you schedule your crops across that to keep your risk the lowest. Now, it won't be the only thing that goes into that factor. There's soil conditions and which paddocks can handle winter and summer. There's a whole heap of things that will also go into that decision. But this can be an important part for some diseases of that decision-making process. For some diseases, you may want to have a range of varieties of some which will have greater tolerance than others. So you can basically put the odds further in your favour by putting the varieties with the greatest tolerance into the higher risk paddocks if you believe that they can handle that. So that's just basically getting the odds better in your favour, because this is really all about probabilities. Now, in the longer term, you can be using this technology to monitor whether you're actually getting your situation for a particular pathogen and disease risk is getting worse or better, and whether the things that you're doing are actually improving the situation in the longer term. So that can be your paddock rotation. It can also be any cover crops or things that you're putting into that system as well. Now, I'm not going to go into too much detail about although Doris did mention some of them about these, but ultimately you're going to be trying to optimise your nutrition, your irrigation, and any amendments, and getting the odds better in your favour, right? They're things that you're going to be doing. Occasionally, a soil-borne disease will actually factor a little bit more in that, in terms you might want to be drier or wetter or whatever, but there's usually a lot of other crop management things that come into play there, just about optimizing quality of the product and yield. So, and then when you get down to the bottom of this list, now, okay, they may be a last resort, but ultimately, as I said, you don't want crop failures. So in some situations, you have to use some of these techniques potentially to bring a paddock right back. But that, I mean, I'm not going to argue philosophies here, but ultimately you don't want to plant into situations that can't be managed, right? So, and this is part of that thinking process. So I guess overall what we're doing is, look, this type of technology is never going to save every crop every time. What it's about is improving your odds of success. It's about providing additional information that you wouldn't otherwise have, and it's a number, it's a quantified number, so that can inform and allow you to make better decisions, and in the longer term, refine your disease management strategies. Right? So for the moment, I'll move on from the pre-plant risk assessment and move on to, I guess, some more, type of thing that researchers do, but it's also something that's very important to growers, because ultimately, if you're looking to control things or manage things, you need to know which ones you're controlling, and often looking at symptoms, some are easy, some are a lot more difficult to tell. Now, on the top line here, I'll just describe them as cavities of carrots. I'll just say that, right? So you've got a range of symptoms of cavities of carrots, right? Now, what are they? Which species are they? Or are they truly cavity spot caused by Pythium? Well, I guess that's the question, right? And some carrot growers, if there are any out there, might be guessing at them right now. Down the bottom, you've got a stunted carrot with also some protrusions higher up on the carrot. So it's been stubbed off and got regrowth. So we look at it, what is it? And then we got another symptom that's completely different, which is crown rot of carrots, right? Which I'll give you the reason why we've put that on this slide in terms of DNA pathogen testing. I'm not going to give you the actual levels that we measured in these carrots. I'm just going to say that ones where we confirm it, they're high levels. So that, along with the symptoms and other knowledge we have, that they're basically confirming that the first one that you've got is caused by Pythium sulcatum. So you've got high levels in that. So it's a symptom for that. The next one is caused by Pythium viola. We've got high levels of that. So the codes there are our DNA codes to show that we've tested them. The third one is also Pythium viola, but it's quite a different looking symptom, and symptoms vary enormously, right, which can make diagnosis quite difficult. For us, it's very important because we're trying to relate it back to pre-plant. For a grower, it's very important because you're going to have a different management strategy, or you're going to go about things quite differently, whether you've got Pythium sulcatum or Pythium viola, right? So then you've got the next one, which could easily be slated as a cavity spot, but in actual fact, whether it's traditional or through our DNA testing, we couldn't get any Pythium out of that. It's most likely something else, and pretty much will be something else. And then the last one, also no Pythium detected in that. Now, some people will see that and probably think, yep, it's most likely pest damage earlier on in the crop, right? So it's just grown to that symptom. So, but it is very important, and this is quite a powerful technology to help in the diagnosis and quantification of these symptoms. Now, the next set that we've got, first one is Rootknot nematode, right? So we've got quite a few carrots like that from different places that have been tested, and we normally get Meloidigan hapla back as a species back on those. So that's Rootknot nematode. The next one We've tested those carrots, we've tested mini soils in that paddock, pre-plant, post, all things is we do not detect any root knot nematodes at all in that paddock, right? The level of symptoms isn't as high as in the other paddock, but nonetheless, it's you could easily think that maybe that was a root knot nematode problem when there's actually quite a lot of other things that could cause that symptom. Now, the last one is just to highlight the fact of what I talked about before. These tests are specific. They pick up specific pathogens. They won't pick up things that we don't have a test for. Now, in the case of crown rot, we do not have the tests for the suspected targets of crown rot at this stage, right? So we're not going to get a result back on that particular carrot. It's important to acknowledge that. And that's why at the end of the day, for diagnosis, molecular is not the be-all, end-all, right? But it is a very powerful tool. On the other hand, traditional, whilst you can get there, if you use molecular, it can, like once you get to a certain point, it is very helpful in coming to a final diagnosis on things. Now, I'll leave that part of it just to show that, you know, it is very useful in determining symptoms and looking at symptoms. I'll go back to another aspect that people don't think about so much from the soil testing point of view, but it's actually one of the greatest uses of the technology, not only from researchers, but also from growers, agronomists, and other clients, is we can spend quite a lot of money looking at different trials, right, and setting up trials. They're quite expensive to set up. You don't always get results out of trials, unfortunately, because diseases don't occur, right, even though you may have changed things that in the crop that you plant after this may not actually show symptoms, right, because you're trying to work out how much, you know, are you increasing or decreasing your disease risk with these various fallowing biofumigator sorghum, triticale, right? What are we achieving by putting those in? And this was a trial exactly about that for Rhizoctonia. So the crop after this didn't show a lot of symptoms, but what we do know is because we measured the DNA prior to these crops and the pathogen DNA after these crops and the pathogen DNA going into the next crops, we know that these crops actually had quite a substantial impact on that. So it's a very powerful technology in adding that extra information across a field trial. Now, a grower may not set up a replicated trial like this, right? That'd be more unusual. More likely that middle bit where we sprayed half out and left half unsprayed of the sorghum is more likely what a grower's going to do through a big strip through their paddock, right? But once again, it's still important questions. If you're looking at trying to manage something, did you have the pathogen? What sort of levels were the pathogen starting at? Did what you do actually change that inoculum level? Because that's what you're trying to do. On top of that, of course, with cover crops, there's all the other benefits that they provide in terms of organic matter and soil structure, and that's a separate issue. But the pathogen side of it, unless you actually measure it, you can't actually be sure what you're achieving. And it's expensive to have these crops in your system. And they may favour some pathogens and not favour others. So it's just an added level of information that's very useful in your long-term thinking. Now, sorry, as I get towards the end, I guess I'll just sum it up by saying, it's very important to know, like, if you're going to minimise the cost of soilborne diseases, it's important to know which pathogens pose a significant risk. Now, that's something that this type of testing can assist with. As I said, none of these things are the be-all, end-all, and nothing's perfect, but it's certainly going to add some information that's quite useful. And it's information that you're better off to know it than not know it, I can assure you. Now, the next part is you also, to capitalise on that, you have to have management options. Now, this is not giving you management options, but it certainly is a tool that you can use to assess how well those management options are working for you. Right? So that's an important part of it. The last part of it is with soilborne diseases, they are quite difficult. And at the end of the day, unless you know what the risk is before you plant and you're already starting to manage that, then you're behind the 8 ball. Okay, Carl, so that's conclude there.
Yeah.
And I'm
Great,
happy to take any questions.
Thanks for that, Mike. A really good presentation, and we've had a couple of questions come through already, but I just thought we'd hold to the end there for the summary. And I think, you know, if you can't measure, you can't manage is a nice message to leave that on. So
yep.
Over to the group now to put forward your questions. We've got one through from Carl Young, Mike, just asking, for the pre-plant test, can you actually detect multiple species in one sample, or is one species needs to be detected per sample?
No, our technology is multi-pathogen. So whatever we can put in a test panel, for instance, we're running panels of 26 different tests on the one 500 gram sample, or even the one tissue sample. Right, so we basically construct our panels up. So within this horticulture project, we're using panels of around 22 different pathogens. So if you were to do a root sample, you might get Rhizoctonia, one of the AGs of Rhizoctonia back. You might even get several species of Rootknot nematode, because it's not uncommon to have more than one species in that. So you'll actually get that breakdown in the one sample. So it's just one sample, multiple target test.
Okay, great. Thanks for that.
We have to have the targets.
Yep. Mike, you mentioned earlier the importance of being representative across your blocks and your field. How much testing do you actually need to do per field to get that representative sample, to know what you've got there?
Look, it really depends on your past history, is a big part of it, right? So if you're growing pivots and you've basically been growing them as half pivots or quarter pivots or full pivots, you know, if you're growing them as quarter pivots, they're going to have a very different history. So you need to be really treating each of those quarters separately. And it's the same if you've got irrigation bays, right? If you're planting different irrigation bays as blocks, I mean, that to some degree is segmenting where you're going to do your testing. Now, the bigger your area, the more testing you'll also need on top of that knowledge of crop history. Now, in a pivot, we would never go with really less than 4 tests. And it also depends on the pathogen, right? So some pathogens like Rhizoctonia, which are very, I suppose, sporadic over a paddock, then you're going to need more intensive if you're going to pick them up. And Pythium violae would be an example of that, and Pythium sulcatum are both ones where if you test 4 samples in a paddock, you may well get a couple of zeros, right? So, which doesn't mean zero, it means it's below our level of detection, right? But you need more than one to cover that. So it's cropping history, size of your paddock, and your target pathogens. And it's something we have to put a lot of work into to actually ascertain that. But what I will caution is that ultimately you're measuring a 500 gram sample of soil. So you've got to do a good job of sampling, but you also need to be realistic about how much area that sample can cover.
That's some good pointers. Doris and Mike, one thing you both mentioned was that looking at the whole system when managing soil-borne disease and risk. Doris, I might go to you just on a question we've got around soil biology and where pathogens fit into that when you're looking at tools like fumigation, biofumigation, and nutrition management. Sorry, Doris, I think you're muted at your end.
Sorry about that.
No, that's fine. We can hear you. Great.
Yeah, so the pathogens are part of soil biology, even though traditionally they'll be studied separately from the soil biology. So it was good to see that SARDI is starting to also look at beneficials at the same time as looking at pathogens. Usually what we're finding is higher levels of soil microbiology would give you a better chance of having suppressive soils, disease suppressive soils. So it's certainly good to go for healthy organic matter levels, and if crop rotation, or if crop rotation is not possible, cover crops and mixed cover crops would potentially give you a better mix of soil biology than just single species cover crops. But again, you have to look at your own situation, what you can actually fit into your rotation. So growing the more organic matter, the better, up to a point, and the more you can use rotation cover crops, the better, also for suppressing pathogens.
Mm. Thanks, Doris. And Mike, did you have anything to add to that?
Oh, look, the beneficials in the overall soil microbiology is, I mean, it's a very interesting space. It's a very important space. But from a DNA testing or understanding point of view, it's a very complex space.
Mm.
But clearly the points that Doris made, where you're basically increasing your microbial diversity is going to be, in the main, will be helpful. So, but understanding it is a very complex space.
Mm. And Tony Phillippi's just sent through a question for you, Mike. For the substrate seedling test, what would a turnaround time be for a diagnosis?
Well, it's going to depend on whether we've got the targets for the diagnosis. So what we're really going to tell you is, rather than a diagnosis, I would put it more down as a quantification of the available targets that we got, and whether they're present or not, and at what level. So that's the way we would approach it. In terms of turnaround time, particularly for those types of samples, they are non-standard samples, right? So we have to deal with them quite differently to what we deal with soil samples. So it's, unless you've got a reasonable number of samples, the turnaround time's not going to be great. But if you do have a reasonable amount of samples, then it could. For instance, we run our potato tests now every 2 to 3 weeks, so you can pretty much be guaranteed that you're always going to get that testing done within 2 to 3 weeks. But for non-standard samples and that side of things, it can be considerably longer than that.
Mm.
So,
okay, good to get a sense of that.
Yep.
A couple of questions we've had come through from multiple people on the line is the issue around cost, and I guess it's one of those things that has to be weighed against the benefits of what you get from a risk-based approach. With Predictor PT and Predictor B, Mike, it might be good to use some of those as representational of, or representative, sorry, of the cost, say, of something in veggies.
Yeah, look, both of those services and it's on the suite of tests that we run for those. They're $180 plus GST. That's the price that's set for both Predictor B and Predictor PT. The vegetable suite's likely to be as big a range of tests as what's running for those. So, I mean, ultimately, these are decisions that need to be made in consultation with horticulture, innovation, right? So, and to make the service viable, so that because the other thing is people do want good turnaround times. So that needs to be factored in, how many samples you're going to get per lot of testing, right? But that would give a general indication of where it's likely to be.
Yeah, okay. That's good to know. Excellent. Now, I'm just conscious of time. We've got to most people's questions. Just in wrapping up, I just wanted to mention a couple of things. One, some additional resources that we've got, as I mentioned earlier, on the handouts pane for you to take away with you today in electronic form. But also on the website, we've got a number of past webinar recordings that have involved other plant pathologists and experts like Len Tesoriero and others on specific species of soil-borne diseases. Doris mentioned masterclasses earlier on. We've got the 4th soil-borne disease masterclass in Adelaide this year from the 3rd to the 4th of September, plus the crop nutrition masterclass that Doris mentioned in Brisbane in October. So Jump on to soilwealth.com.au to have a look and register for those events if you're interested, plus access some more resources. And that takes us to our full hour. Our commitment to you today is to get you out of here and back to where you want to go or need to go, out into the field or back to the desk within that hour. So look, thanks very much for participating and coming along. And also a big thank you to our two presenters, Doris Blaesing and Mike Rettke as well. That's been a really informative session. I know I've learned a lot too. And we'll see you at the next session. Thanks, everyone.